I'm going to present this on behalf of the ESA team. This is a product of 2 days of lots of discussions. So what we did, I'm going to report back from the different sessions that we've had, and basically the presentation wraps up the issue, the crop, the identified gaps, the approaches, stroke opportunities, and who the focal points are within our region, and then from the GG program. And the first crop here is sorghum, and there are a number of traits that have been identified. Most of these can be picked up from the gene bank. We are going to screen the mini-core, select collection. Some of these are within the region, some are not yet within there. But Eric is going to work on this with Harry to acquire this germplasm. So drought, striga, anthracnose, and leaf blight, a combination of 1 abiotic and 3 abiotic stresses. And then you have the others where we don't have a solution are marked in red. This is stem borer and shoot fly. This is going to be handled through pre-breeding. Again, it is Eric, Shivali, and HC Sharma are going to work on this, collaborating with Harry to deliver at least the first products, which will then go into the main breeding program. You have others that we have some solution, we believe, in the gene bank. We can screen our accessions, and we have the gold midge and smart, which is another disease. And then we have one related to nutrients, that is iron and zinc. Again, we shall screen the mini-core accessions. For finger millet, most of the traits that we have, blast, striga, and drought, these are related to input traits. They are production-related issues. And the only one that is iron and zinc is nutrient. Stiff stalk, this has to do with production. So again, most of these are going to be screened from the mini-core collection, and Henry and Harry are responsible. These materials are going to be acquired from here if they are not yet within the region. Groundnut, we have the usual culprits. We have drought. We have 2 diseases, the leaf spots, and then we have leaf miner. This is an emerging issue. Again, we believe that most of these can be found within the mini-core collection, so we shall just be screening those materials. And then pigeon pea, we have fusarium wilt, we have sarcospora, 2 fungal problems, and then we have the issues related with nutrition, high protein and calcium and iron, and then we have the vegetable type, which has to do with when you use the crop, and then we have the red, the one marked in red, pod borers and the pod fly. These we don't have material within the gene bank, and again we are going to find solutions for this through pre-breeding. So Gangarao and Harry are going to handle all issues related to the mini-core screening, and Gangarao, Shivali, and Harry will work on the pre-breeding activities. Chickpea is going to be led by Chris and Harry, and there are again a number of activities a number of traits that we are looking at, again related to input traits like ascochyta, early maturity, mechanical harvesting, herbicide tolerance, drought and heat, cold and frost, and the one which is output related is the vegetable purpose again. And then you have the 2 marked in red. These are the ones we do not think have material within the gene bank, and Chris, Shivali, and Harry are going to work on the pre-breeding. For pearl millet, the issues are really iron and zinc, and again it's about screening the mini-core. These are the issues related with the gene bank and pre-breeding activities that we are requesting the RPGG to be able to support the ESA region breeders in implementing. Now, the next part of the presentation, you can ignore perhaps the top there, the template was the same, is the issues related to forward breeding, gene discovery, cell and molecular activities. We identified only one groundnut that we do not think we have a solution for, and we're going to try to use transgenics for this. So it is myself, Babu, Haile, and Pooja who are leading these activities. The rest of the activities are at different stages of either forward breeding and or gene discovery, and that includes drought, oil content and quality, iron and zinc, and then the foliar diseases, that's rosette, leaf spot, early and late, rust, and then we are going to do something else. This is the early generation screening. We are going to be looking at mainly the diseases that we have. We have about 30,000 lines that we're going to be sending here. And Manish will be working together with me. And we are going to be sending this within this year. Chickpea has the same activities. Pod borers, we don't think we have a very good solution. So again, Pooja is going to work together with Chris and Damaris from the region to deliver materials that may provide solutions to this long-standing problem. And again, dry root rot. Now, early generation screening, there are about 3,000 lines from Chris and Gangarao, 1,500 or thereabout from either party. And they'll be working with Mahinda, Chris and Gangarao. The same is with drought, heat, frost, and Ascochyta blight, I believe, is AB. So these are the issues that are available, except I believe for Ascochyta blight, that's where the gene discovery is going to be the main focus of the activities. I don't know, I left my glasses. Pigeon pea, the culprits are again pod borers and a pod fly, and this is where we think genetic engineering may provide us a solution. So Pooja working together with Gangarao and Damaris are the leaders in this. And then you have drought and the maintenance for the hybrids. This is mainly going to be work within gene discovery and trying to validate markers. So it's Gangarao, Damaris, and Rachit who are, oh, thank you very much. Okay, so sorghum, there is no early generation screening for pigeon pea for now because most of the work is within that range of the markers are still being validated anyway. So for sorghum, we have 3,000 early generation lines that are going to be screened, and it's Eric, Santosh, and Damaris who are going to be involved. Again, these are going to be sent within the year. They are already planted, and so maybe they will miss the first crop, but the second one they will be able to supply. Drought and stay green, there are markers available for this. So again, Eric, Santosh, and Damaris are going to be engaged. Iron, zinc, should fly. These are going to fall within the gene discovery portfolio of activities, and it involves Eric, Santosh, and Damaris. Striga, this is a long story, but we agreed that we should have cell and molecular work as much as possible, gene discovery, and where possible, we'll try to screen the other materials available, and it is the same group of people that are involved. Finger millet, drought, striga, blast, iron, calcium. Yeah, for most of this is about gene discovery and to an extent cell and molecular activities, and they will involve Pooja, Santosh from here, and Damaris and Henry from the region. Henry is the breeder in this case. Pearl millet will be the same. This is where we have some markers that need to be validated, and all QTL have been mapped. And so you have drought, iron, calcium, and zinc. And so you have Henry, again working together with Santosh and Damaris. And for drought particularly, there are some markers available. And so in this case, it's just going to move forward to forward breeding. I think the last slide is about yesterday's afternoon session. This is what we're expecting. The region is requesting from Abhishek and his team. It's about operationalization of BMS, especially within ICRISAT community, including our partners. There is the whole issue about data management, curation, analysis, archival, and sharing support, making sure that that happens in earnest. And there was someone that we had a very long debate. It's about Abhishek spending more time in Africa. There were issues about numbers, 30 to 40%, but for political reasons we decided to remove it. I was told in Africa the voice of the people is the voice of God. So we removed it. This is a bit busy, but this is the output from yesterday on the seed systems. We looked across a number of issues. On this right, the blue panel, the key areas ranging from production and partnerships, seed marketing and distribution, capacity, awareness creation, and policies. It was done in 2 separate sessions. For cereals on the first day, and yesterday for, I think this morning for grain legumes. And so the issues we found are very, very similar, and I'll just give you a flavor of what's going on. Under partnership, we noted that both formal and informal seed systems are working, but we need to dive in a little bit deeper to gain deeper understanding of how they really work in different types of communities. The big issue here is about early generation seed production, breeder and basic seed that is critical for both of them. The approaches to deliver that is mainly through civil society and farm organizations, as well as public and private sector engagements. There are a number of lessons and the entire team that's going to be able to work on this from the ESA program and from the GG program are listed, and that list runs the whole way down. There are issues about marketing and seed distribution. It's all about participation of private sector. They all relate more to market structures, weak market structures that affect access and use of our seed. And again, they're highlighted, disconnection between seed supply and demand, the fact that our seed sometimes is very pricey and they are skimmed off the market before we can go after, and so on and so forth. So we think that having formalized seed maps and really thinking more about how our formal and informal seed systems work is extremely crucial. And we also think that there's a seed harmonization mechanism going on within the two trading blocks that we have in our part of the world, the East African community and the common market for East and Southern Africa, also called COMESA, that have set up seed harmonization to the extent that when you release seed in one country, it can in two countries, you can send it to another country without necessarily having to go through further testing. Capacity, there are a number of issues ranging from the recall systems to the others that are not written here. I'll just present what is written here. But we think that having quality assurance systems that are going to ensure all the way from production to delivery will help us to deal with the whole problem of adulteration, financing mechanisms, including among others the seed revolving fund as practiced on and through ICRISAT over the last 7 years could provide something that we could learn for up and out scaling to other regions. Awareness creation is another one. There are a number of issues. We think that there's not enough communication about our legumes, particularly to potential government authorities. When they talk food security, our crops are really not there, and we need to get them discussed in higher level boardrooms. There is the whole thing about the way we are messaging about our crops, that sometimes we are just too negative about what we say. We need to communicate the potential dangers of our crops, but also the bigger benefits that are there, because that eventually affects the demand that there is for our crops, and aflatoxin is one of them. We can't continue saying groundnuts, you eat them, you're in danger of death. Okay, there are many ways of awareness creation, and these are some of the portfolio of activities that we are doing. Among others that we have not done here is high-level support from our leadership, talking to some of these big boardrooms. We think that would help. Okay, policy, there is the institutional strategy for seed systems is still under development, and restrictive seed certification systems, we don't have really very good monitoring systems for our release varieties. When do we actually manage our how do we manage our varieties post breeding levels, and make sure the replacement cycles and so on are well studied, the market structures, etc. And then there are issues about variety release. All our varieties are released through the public domain, and then the inadequate government intervention in both legume and cereal seed systems. So there are a whole shopping list of things we identified, and there are also issues that we think we can contribute to, and this particular slide summarizes most of what we need to do. There is detailed need. So bullet number 1 about developing a strategy that is implemented by the institution and partners is critical, and all these things reflect some of the things that we need to include in that part of the strategy. There is a draft strategy which Emmanuel, the theme leader, has. I think that's it. The only other issue that was remaining was raised by ECPO. It's not inside here. It's off my head. If you can vocalize. Where is he? Yeah, could you say it's a give it mouth? Yeah, that's the one that I missed out. Yeah. I hope this captures most of what the team discussed. And yeah.
Trade discovery and forward breeding summary. Mahinder will be responsible for this genomics and trade discovery related issues, and Manish Rudkiwal will be associated with the forward breeding related things. So in that summary table, there is only just forward breeding. If we have that genomics and trade discovery and forward breeding, we can add Manish there. So there should not be any confusion.
Okay.
Yeah.
No, you help me, and you. I think Gangarao has the whole document. We just correct it with him. No, it was deleted after some lengthy discussion. Maybe Gangarao can explain why it was deleted.
We discussed, and if we can do screening, simple means, that's what we discussed, and we have removed that one. If it is, we have a large number of populations, then we can take the help of the fusarium through markers. If it is the population size is less, then we can do the conventional breeding. That is what the group discussed, and then we removed that. But still we can put it if we have the markers, they are very reliable.
Yeah, so what was the idea first when we were discussing? It was to, whatever the markers, what we got for fusarium wilt, first to get the feel, the validation in the African germ class what we have. So that was the basic idea rather than directly using in the breeding program. So that was kind of validation rather than using directly into the breeding program.
Yeah, then we can.
So that was the idea.
Put it as for the validation,
yeah,
and also look for the African land races,
right,
the pathotypes.
Right.
Then that is.
Okay, Patrick, I've got a question for you, and you'll give me a critical, frank answer. So, you have a meeting like this, you have these plans put up here with responsibilities for people, but will anything change from, because of this meeting and those plans, from where we were a week ago? The reality is we've got to align with current projects and deliverables. We have our funding. We're in almost June in 2016 and we're doing planning, so people have already know what they're going to do this year. So has this meeting either modified, changed, updated, created new work plans for people? Has anything changed because of this meeting and all that planning you put up there on the board?
Oh, okay. I don't know whether I'm authorized to speak for and on behalf of the others, but I'll speak on my own behalf. Yes, on my part, I think some things will be modified. They are added on. But there are others that, because of the nature of our cropping season, it's already, it's rather late. They will start towards the end of the season. So the meeting by and large has been beneficial to me, at least, because we've been able to have very frank and open discussion with our colleagues here. So the things that are easy to go, the low-hanging fruits, I think we'll be able to implement those. Yeah.
Well, I've got 5 worksheets here of all these things you're going to do with names against them.
Yeah.
Does that mean they're just going to sit here in the record of this meeting, or is it actually going to feed through into work plans and what people are going to do and aligning current and future projects and proposals against what we said we're going to do? I mean, I know it's unfair asking you, but that's a question for the room here. Does this meeting change what we're going to do or modify what we're going to do? Going to throw to Moses? What do you reckon?
You want an honest answer or a politically correct answer? I think to begin with is to get the genomics people to stick to one terminology. They are promised as markers. They shouldn't come back and tell us SNPs and start telling us other stories. So they promised us markers. We want markers from my team. I think that for me it has been useful, and I'm sure it's going to change the way we work, because at least for me, for the first time, I know exactly what Puja does. I had no idea. Just hear her talking in tongues, a language I don't understand. So I think it is helpful that it has put into perspective, I think for my team as well, what we can get from them and what they can get from us and what we can work together. So I'm very confident that it's going to change the way we work. Regarding the way we have looked at it and put the names against certain activities, I'm hoping the money will come from your pocket, the CRP money. So we have made your job easier now, where to put the money when it comes to genetic gains. You can fund us along those lines. But more importantly, I think that if we get opportunities to write proposals for funding, then this is the framework in which we should be doing it, because these are the areas we have distilled in terms of priority and identified the people who should do the work.
Yeah. But I think also that there are parts of this presentation that they represent activities that we were not doing before because of obstacles, institutional in nature. For example, with the gene bank and pre-breeding activity, where we needed to access accessions from the region and from here to the region. So those types of activities have been part of the traditional breeding program, but now we will be able to activate them and run along with those. Now, I think the other difficult question which we have to deal with, and I think we've discussed this with Rajiv, we will need to find mechanisms of operationalizing particularly the forward breeding and gene discovery component. How we work together with the colleagues and let me say capacity in the region and the capacity here. This is something that the discussions have been on, and I think they'll continue from here on after. So those are the two issues really.
Okay, thanks, Patrick. We've got two other presentations. Maybe I asked my question too early, but I'll ask the same question to the other groups maybe. I mean, because that's what we want to get out of a meeting like this. It's not just sit for 2 days and have a talk and then go back and do what we're going to do, but how do we move our agenda forward collectively? And it's really good to see that there is that positive attitude in the room. Any other questions for the Eastern South Africa? Shivali.
Patrick, can you see the list of the trades, please? Patrick, would you please bring slides of the list of the trades? Because while discussing,
Where exactly do you want us to go to?
Groundnut for pre-breeding.
Yes.
Yeah, because we discussed about the
Yeah, just go ahead. This one?
maybe I have missed it somewhere, or I just want to see whether I missed it or somehow it got missed. See here, especially for leaf minor and late leaf spot, we discussed about the pre-breeding intervention, but I think it is not being reflected here. So maybe if the trait was dropped out later on, so would you please
Explain. Yeah, so we had a lengthy discussion in the team, and they felt that going for pre-breeding is a long shot, and that these materials are probably within the germplasm. So that's why we dropped it. Yeah, that's the explanation.
Any other questions?